I'm very pleased and honored to have Michael Strano from MIT. Michael and his team are part of our center here, and the primary role, in addition to developing methods to understand the nanobio interactions, is to help us to precisely engineer emerging 2D materials that and make them available to the nanotoxicology community. So for all of us in the nanotoxicology field, it's always this elusive issue of protein corona. So we know that particles in biological media, they will absorb all these proteins, and it's kind of the chicken and the egg paradigm. So it's the intrinsic properties of nanoparticles will define these coronas, and we do know now that these coronas also affect the nanobio interactions, cellular uptake, etc. So Michael and his team, they're really leading this field, and not only understanding the corona formation in various media, they are trying to engineer the coronas. So which is that opens up a number of applications for biosensors and some other applications in agricultural areas as well. So I will keep my introduction to Professor Strano's very short. As a long CV. I want to congratulate him because very recently, I think last week, he was admitted to the National Academy of Engineering. Congratulations, Michael. It's a great accomplishment. He's a prolific writer, recipient of numerous awards, and also very recently was named by the Thomson Reuters as one of the most influential scientific minds. So without further ado, Michael, I will give you the podium here and look forward to hear your great research.
Great, thank you. Thanks a lot. Thank you. My pleasure to be here today. And, are you okay? Okay, good. So I thought what I would do today. First of all, it's my honor to be here today. I'm a chemical engineer by training at MIT for about 9 years. I lead a research lab at MIT on the chemical engineering of low-dimensional materials. We have about 25 postdoctoral and graduate students, and we work in a variety of different areas: energy, biomedical applications, materials. I just wanted to talk today about relatively recent work where we've been interested in just given the department and given the interest of this part of our group. I thought I would focus on the nanoparticle corona and increasingly focused in our work as we try to make nano devices that operate in the human body. And I'll also talk about some emerging work that we have on plant systems in vertebrates. But I thought I would start off for this audience. I hope I'm making the right decision. I thought I would start off with a tutorial on particularly carbon nanoparticles. So most of my work, about 70%, is on carbon nanotechnology. I thought it would be a good way to get started. A short tutorial that emphasizes a couple of things. We're still in the early stages of understanding the electronic structure of these different carbon nanomaterials, how it impacts their biological activity and their structure and function. So just to get us all on the same page. So we're all familiar with graphite, the graphite in your pencil from general chemistry. The three-dimensional material, the three-dimensional crystal. If I exfoliate it to a single layer, it's called graphene. So graphene, and the idea is that the electrons in graphene are literally smushed into two dimensions. Graphene is not the same material as graphite. It has very different properties. And if I roll graphene into a cylinder, I make what's called a single-walled carbon nanotube. So very different electronically, chemically, optically, very different materials. And I think that graphene is the only material that has this very unusual property that all I have to do is change one set of lattice bonds and give it a helical twist or exfoliate it into a single layer, and I really get a completely new electronic material, almost like a new element. I won't talk about how carbon nanotubes and graphene are grown. There they're made in a variety of different ways, anywhere carbon condenses. For example, carbon nanotubes are grown normally by chemical vapor deposition for, over a catalytic nanoparticle. I mean, the idea is that are produced worldwide at the thousands of tons per year. So they're becoming like a commodity chemical. So I think they need to be treated in that way and become the subject of a lot of the pioneering work that has been done here. It's very important to understand their environmental and so forth. Carbon nanotubes, for example, they can grow like grass on its surface and vertically aligned them, or you can cut them very, very short. They can be as small as proteins, like 10 nanometers. And so many materials have this kind of morphology change, but carbon is unique in that depending upon how you roll this graphene sheet, you can make fundamentally new electronic particles. And this is probably new to maybe part of you in the audience. That depending upon how you roll this graphene sheet, you make these structures that are both metallic. I'm going to try to use the pointer here. Remember, we have people under that. So depending upon how I roll this graphene, I can make nanotubes that behave like metals. They'll conduct electrons. Others will be like semiconductors, like the semiconductors in this computer. And then others are more characterized as semi-metals. But there's a huge diversity. It's unusual in that it has this property. And I want to take a few slides to really illustrate why this is true, just so you understand. Graphene is a very unusual material. It's a two-dimensional material. It's called a zero bandgap semiconductor. And you can plot this electronic structure. This is called reciprocal lattice state. So this is how physicists and material scientists will look at the conduction band and the valence band. So you can probably remember this from general chemistry. So graphene has this six-fold symmetry, and graphene looks like a metal along 6 points in its reciprocal lattice state. So if I look at graphene along these nodes, we call them nodes, kind of floppy nodes. I'll call it floppy nodes. So if I look at graphene along this critical graphic axis, the conduction band and the valence band touch, so it looks like a metal. And then if I look at it off axis, it has a semiconducting gap. And this unusual feature of graphene is what gives rise to this huge diversity of electronic structure. Okay? So if I map this, it's called the electronic dispersion relation. Map this onto 2D. So this is that function mapped onto 2D. You can see what happens if I roll a graphene sheet. Literally, what happens in a graphene sheet, circumferential. So the electronic wave vector now is constrained with integer multiples of the circumference. And that means that 2D electronic dispersion is now 1D. It's pretty simple. Function intersects one of these metallic nodes. The tube is a metal. It behaves like a metal, conducts electrons like a metal. It's a lot like a metal. Pretty electrons. But if I roll it in a different way, on axis, say, I get a semiconducting band gap, and that's a fundamental change in the electronic structure. Further here, all right. I can hold it like this. How about that? What if I do that? So that way, if I turn, is this better? Online crowd?
This just means my hands are full the whole time. Okay, no problem. I got to juggle all this. All right, so this comes up. So there's a vector that connects the 2 points that go into rolling. Literally, it's the number of hexagons. So this is called the 10-10 tube. These two integers. It's 10 hexagons along the two basis vectors of graphene. So I roll this graphene sheet and I make this. It's called the 10-10 tube. It belongs to a class called armchair. This comes from carbon science. You're supposed to see here. Whoops, I got to remember. You're supposed to see here. You're supposed to be sitting here. This is a chair, and your arms are sitting here. in the chair. I didn't name this, but it's like you're supposed to see a chair here, and you put your arms here. Anyway, armchair tubes are always metallic. They behave like metals, and if you so what chemists and physicists, we think of solid state materials, we map what's called their density of electronic states. So this is how many electrons like to reside in the material at a certain energy. And for a one-dimensional particle in a box, you get these sharp singularities. These are called Van Hove singularities, and they're characteristic of a one-dimensional system. And so they're present for carbon nanotubes. This is a metal, so there's some electron density that connects the valence and the conduction band. So that's a 1010 tube called armchair. And you can roll the tube in a variety of different ways. Like this makes a 10-5 tube, 10 hexagons, 5 hexagons. And the 10-5 tubes it's a misnomer, but we call them chiral tubes, actually. So, and they're metals or semiconductors if these two integers, n minus m divided by 3, is not an integer. Then it is a, if the mod of n minus m3 is not an integer, then it's a semiconductor. Otherwise, it's a metal, and it's called a chiral tube. It also has these Van Hove singularities, and you notice this 10-5 tube is a semiconductor. It has a gap, so it has a band gap, and these Van Hove singularities dominate the optical, chemical, electronic properties of these materials. They're very distinct, and you can see here there's almost a new periodic table of materials. So when we talk about carbon nanotubes, we have to remember there's a huge diversity of electronic structure here. Roughly, the red tubes are metallic. The black entries here are semiconducting, and their band gaps are fundamentally different, from small band gap to large band gap. Generally, the diameter is decreasing, and so I'm trying to convey to you there's a huge diversity of electronic structure here. Okay. Semiconducting carbon nanotubes fluoresce in the near infrared, a property that I discovered as a postdoc at Rice University, and then one that I've used as a diagnostic and sensor tool, and I'm going to talk about that today. So what do I mean by that? These are carbon nanotubes dispersed in solution. I'm using a laser pointer. Normally I'm talking and I have a laser pointer, but let's pretend that I have a laser pointer. I'm shooting it in through this solution, and then just with an off-the-shelf digital camera, you can see this near-infrared fluorescence that's emitted. And what's happened is here I'm exciting in the visible. So this second band of Van Hove singularities, I'm exciting an electron from the valence band to the conduction band. It's decaying to the band edge, and then I'm seeing fluorescence across the band gap, which is in the infrared. And there's a now famous experiment that we do routinely. I can vary this laser pointer energy, and as I become resonant with those two, with the pair of Van Hove singularities, I excite different carbon nanotubes in my sample. So even though that sample had 40 different carbon nanotubes, I can actually do what's called an excitation profile. I can vary this laser pointer energy as it becomes resonant with different carbon nanotubes. I'll see fluorescent emission along this infrared wavelength. Okay, and so you can see even now there's a variety of different chemical species that we can use, all indexed by this chiral vector. Okay, this fluorescence is has very interesting properties. First, it's completely non-photobleaching. So many of you in the audience, if you have a biological background, you have used fluorescent dyes for staining. Most organic fluorophores will photobleach. Carbon nanotubes have no photobleaching threshold. This is indocyanine green. It's a near-infrared fluorophore that's used. It's actually FDA approved for biomedical imaging. But see, it photobleaches readily without too much light. So this fluence, and under the same conditions, you'll get a pretty steady fluorescent signal. So this told us early on that we can start to think about nanotube fluorescence as a stable sensor, a sensor that may operate for days, weeks, months, even years, something that we've recently realized. You can have an optical sensor and have it be stable for literally years. It's very few materials have this kind of photostability. You can really pump a lot of light into them, and it has to do with the fact that there are no dangling bonds. And then this near-infrared emission is uniquely situated in the tissue transparency window of many biological matrices. So the human body, for example, is not transparent to visible light. Blood is the dominant absorber here, but there's a window where between where you can get blood and water absorption, where you can get about 4 to 5 centimeters of near infrared light. And there's a whole area of biomedical imaging, a lot of which has been pioneered here at Harvard, where you can use infrared fluorescence and spectroscopy and autofluorescence to probe deep within the human body using very efficient optical means. The early part of my work, I pioneered the study of how molecules can fundamentally change this emission. Carbon nanotubes are no different from other fluorophores in that they respond to their environment. There's something called subatochromism, which we've studied and have a theory of. Other molecules like charge transfer and things can quench the fluorescent emission. There are a variety of different mechanisms that you can use where a molecule can absorb to the carbon nanotube and change its emission. And I'm going to talk about that in my talk. So these are the three topics that I hope to cover. If I run out of time, I'll just go through the last one quickly. But I wanted to talk about a new technique that's come out of my lab where we've been engineering corona phases for molecular recognition. And I'll tell you why that's important. And then I thought particularly for this audience, we've done work where we've been making sensors that can be used in vivo for very long-term monitoring of biomarkers and different things. So we'll talk about that work. It's a new area that we've pioneered just in the past few years. And then I've also been very interested in plant systems. We've made some important discoveries here, and we now understand how nanoparticles move and traffic in plants for the first time. And we have a lot of crazy ideas on what to do with plants and how to engineer them and how to give them new and unusual functions. Okay, so let's talk about corona phase molecular recognition. Part of my lab works on sensors, and so if you want to make a chemical sensor, a good starting point is to have a molecular recognition site. And unfortunately, when you look at molecular recognition, there are very few general platforms to recognize molecules. Almost all of them come from nature. So even in our biomedical assays that are removed from a medical from a living thing, antibodies are still the mainstay for recognizing molecules. There are also these molecules called aptamers or adaptive oligomers. So these are DNA or RNA sequences that fold and recognize an analyte. And beyond antibodies and aptamers, there really isn't much else. There's a niche technique called molecular imprinting where you make a polymer, you polymerize it around an analyte. It works for a very, very narrow set of analytes, and it really makes an absorbent. It doesn't make a recognition site. It's really an absorbent. So we entered the field. There's a pressing need for synthetic and stable non-biological recognition sites. So these antibodies and aptamers, natural systems make them with the idea that they would degrade over several days or weeks. They're not meant to be long-term stable in aqueous environments. Okay, so this is our discovery. So we call it corona phase molecular recognition. And I believe it's a very generic phenomena that we should look for in a variety of nanoparticle systems. So carbon nanotubes are like many nanoparticles, they're hydrophobic. So if you want to put them into water, you have to absorb a surfactant or polymer layer. So this adsorbed layer is called a corona. You're supposed to see the rays of the sun here. And the misconception in our field has been that these corona phases are necessarily disordered, like disordered noodles, right? So noodles can't recognize anything; they're random, right? However, if you do the following empirical experiment, I'll show you lots of evidence for this. If you do the following empirical experiment, if you make a compositional library of polymers, let's say you make 100 polymers, they vary in composition. You form 100 different corona phases around carbon nanotubes. Then you can use this near-infrared fluorescence to quickly screen their response to an analyte library. And if you do so, you will observe that some corona phases that otherwise have no connection to the analyte will show a very selective optical response, either as a fluorescence change, like a fluorescence quenching or brightening, or as a wavelength shift. We call that somatochromism. And you can screen this and look for this very, very quickly. So we call this form of molecular recognition Corona Phase Molecular Recognition. It's not just grafting antibody. It's not at all grafting antibodies or known recognition sites to the surface of a particle. The recognition comes about from the corona phase, and I'll show you examples. We have a lot of examples of this now, and I'll try to make this not tedious. But perhaps the most surprising is this molecule. It's called a Ritzy PEG. Ritzy, we take a diamino PEG and we put these two fluorophores on. This makes an anchor loop structure. You can see this in molecular dynamics. You see the hydrophobic Ritzy sticks to the tube. It makes these loop structures that stick off. So this corona phase will recognize estradiol. It doesn't distinguish between alpha or beta, but it will recognize in that it forms a quenching response with 17 alpha estradiol. And if you mutate the corona phase, your selectivity goes away. In fact, it starts to look like just a lipid PEG structure. So we call this recognition, which emerges from the corona phase, as corona phase molecular recognition, and it is quite interesting. It requires an explanation. This is in the same paper we looked at. This is actually a single molecule near infrared visible turf microscope, and you're supposed to see here. This is the infrared signal of a nanotube lying on a surface, and this green is actually a dequenched Ritzy fluorophore absorbing from the surface. Okay, so you can actually see this detection happen in real time, and you can show at the single molecule level that the system is recognizing through the corona phase, through a rearrangement, this estradiol. You can actually see this and study this at the single molecule level. Okay, I have a lot of examples of this. I'll go through, and then I want to talk about applications. This is we also talked about in our original paper. This is an FMOC branched PEG. It's a PEG brush, and it also forms a variant of this loop type structure. These FMOCs are a common protecting group, but they're aromatic. They're hydrophobic, so they sit on the surface. So this 8-member will detect L-thyroxine, but not 4. So if you change the number of loops, then the corona phase changes, and you can detect L-thyroxine, but not. But then your selectivity decays if you change the structure, or the N group if you aminate. So you're seeing that a mutation causes a change. And this is another example. This is riboþlavin. This is now looking at a wavelength shift, which is much rarer. But this phenoxylated dextran. This is a dextran that we phenoxylated. We put a boronic acid on, and it will recognize riboþlavin, but not FMN and other pendant diols. And you can see here this wavelength shift is robust enough to even see in live cells. So immediately these nanoparticles are useful as cellular imaging probes, and I'll show you that a little more further in the talk. But you're supposed to see that there are these recognition elements. We started to produce some predictive models. So this is a mathematical model that we have to try to predict how an anchor loop corona phase can recognize a certain molecule. We've modeled this anchor loop by ignoring the loop and just assuming the anchors. So we basically looked at we modeled this as a 2-dimensional equation of state of just the anchors. And so the anchors themselves, they adsorb to the interface. And then we developed an equation of state model that takes into account several contributions to the free energy of the system. You can account for the entropy of the adsorbed molecules on the surface. You can account for the excluded area, and then also these interaction terms as the analyte interacts with the anchors and so forth. And you can relate those to the bulk phase compositions, and you can solve these equations. And these energetic parameters you can actually get from DFT and other computations that involve just a simple force field. And I'm going to skip a lot of that, but it would show you that the model has some predictive capability. First, the model can predict the adsorption isotherm. So you see this here. This is experimental data. And the 2D equation of state model does a pretty good job of describing the the adsorption isotherm of this is L-thyroxine, this is estradiol. So you see, it does a pretty good job of describing the dynamics of the thermodynamic equilibrium on the carbon nanotube. And then this is a massive predictive model. It has the model does a good job of predicting what can't be detected from our library. There are some false negatives. There are molecules predicted to adsorb, but the carbon nanotube doesn't optically respond. And then where the model works, it's able to, with reasonable success, give a dose-dependent response. So we consider this we introduced in about 2013-14. It was a pretty good contribution to being able to predict. This gives us a very powerful tool to make new sensor interfaces, and we're still playing around with this. So I had Sebastian Kraus. He was interested in neuroscience. So he did a CoPhMoRe screen. This is an example of what a CoPhMoRe screen looks like for a panel of neurotransmitters. And you can see here, this is, so he found a corona phase that's pretty selective for dopamine. It's not perfect. It actually has some cross-reactivity with epinephrine, but this paper sailed through JACS, actually, because actually we were able to claim, and it's true, it's the first optical sensor for dopamine. So the chemists they never got around to making a fluorophore reporter for dopamine. It's such an important molecule, right? But it just never, so you can put carbon nanotubes on a surface. That's what you're seeing here. And Sebastian is just dosing 1 nanomolar solution of dopamine. You see this very bright turn-on response. It's a very unique, and it's very reversible. You can see this here. It's very fast-acting and reversible. Remember, just 1 carbon nanotube on a surface is an addressable nanosensor. So this motivated a now famous experiment in the group. Very, very exciting. We just published this in PNAS. And what Sebastian did afterwards is he put an array of nanosensors, which by the way are sensitive down to even single molecules of dopamine. So very exquisitely sensitive nanosensor platform. You're supposed to see here there's just a large array of nanosensors underneath a PC12 neuroprogenitor cell. So these cells, if you stimulate them with potassium, they will efflux dopamine in a very fast concerted event. So here we basically show with about 20,000 nanosensors underneath, you actually can see the dopamine release from the simulated action potential. This is not simulated data. This is actual experimental data. You're actually seeing in real time the spatial and temporal dynamics of dopamine release in and around a cell. It's an example of what you can do with a nanosensor that you can't do with other platforms. You can see in space and time where dopamine is being released around this cell, and where the maxima are. And so in our paper we did this for a large number of cells. You see these hot spots at maximum. This is where the maximum dopamine release. And in our paper, we were able to answer basic questions about where dopamine release is actually maximized along the surface along the periphery of the neuroprogenitor cells. Actually, quite surprisingly, we found that the regions of greatest curvature are not where dopamine is released. It's you can see this here. This is the curvature. This is a summary of all of our cell data. This is the curvature, and these are the maxima where the dopamine happens to be released locally. You're supposed to see that the mode in this distribution is actually shifted towards the negative curvature side. So it's these regions right before the points of negative curvature. The point here is that you would never be able to answer this question without an exquisitely sensitive nano imaging array. It's almost like a chemical imaging device that's non-invasive, it's label-free, and you can image it on the surface. We've made some progress on CoPhMoRe. This is a paper that came out about a year ago, and it documents that you can do CoPhMoRe on proteins. This is I had a student interested in we're working on insulin sensing, and we actually have found a CoPhMoRe insulin recognition site. But this is some of our early work here. The student Gilly Bisker, she took the 14 or so most abundant proteins in human whole blood, and she added insulin, and she did a first round CoPhMoRe screen. These are different corona phases around the carbon nanotube. She found this exquisitely selective sensor for fibrinogen. In fact, it'll recognize fibrinogen even in the presence of albumin. Albumin, if like some of you know, is very sticky. It absorbs to interfaces. Anyway, this fibrinogen is an interesting molecule. It shows up as this dumbbell structure. You see it has these three nodes. Anyway, unlike a small molecule like estradiol, it's very easy to show that the fibrinogen is being recognized. And actually, we chopped up the fibrinogen and showed that it's actually the D1 domains, these dumbbells on the ends, that actually are being recognized by this corona phase. But the key is this corona phase otherwise has no connection to fibrinogen. It's really a new area of molecular recognition, and we also showed that this can work. You see that this is the response curve for different chirality carbon nanotubes. You see that the KD is very dependent upon the diameter of the tube. These have different diameters, and so we showed that the corona phase is altered even by the diameter of the tube. This just goes to show you some of the power of this technique. We're writing up this paper now. This is a corona phase that can recognize carbohydrates. We're writing a paper on carbohydrate recognition. This is just supposed to impress upon you how powerful CoPhMoRe can be. Probably the most difficult molecular recognition problem you can think of involve carbohydrates. They're notoriously difficult to selectively absorb. So this is an example. This is a corona phase. This is dulcitol, sorbitol, mannitols. Anybody know what these are? These are the sugar alcohols that flavor your mints and your gum. So probably you have consumed one of them. But if you look closely, you really have to be a chemist who looks closely to see that they really are different molecules. They're six-membered carbon sugar alcohols. Their compositions are identical. They vary by very subtly. I mean, just the one OH group, cis versus trans, but they are different, and it's a very difficult molecular recognition. So Jiyoung An, who's getting ready to graduate, she showed that this CoPhMoRe phase, if she puts the boronic acid group meta or para, she can actually distinguish between dorsitol and sorbitol, and that is a very difficult from a molecular recognition standpoint. I believe it's the only synthetic molecular recognition platform that can do this. And if I had more time, I would show you. We actually have recognition sites for sucrose, but not glucose and fructose, and also we've been able to detect L-arabinose actually over ribose and D-arabinose. And so it's a powerful technique. Let me see how I'm doing on time. Okay, so I wanted to talk about platforms for in vivo measurement. This is something that we pioneered in my lab, and this makes use of the fact that the carbon nanotube is actually a very stable near infrared fluorophore. We have very few fluorophores that operate in the infrared. So if you look at tissue penetration, because of the way scattering and absorption in the human body works, visible fluorophores can barely get with a couple microns into tissue. If you go to the infrared, you have indocyanine green or quantum dots. You can go out to further wavelengths. I'm being generous here, but their photostability, I already showed you before. You're limited by photostability. So these have mainly been used for imaging applications. I put here for carbon nanotubes, I put years, but really there's no photobleaching threshold. So you're supposed to see here with very simple optics, very little, there's plenty of light to detect some and to do a reporter like that dopamine experiment. We can do that in vivo. And one of the top topics, when I first moved the lab over to MIT in 2007, there's an active group, Jerry Wogan, probably you people know, and Steve Tannenbaum, Bill Dean. There's an active group, very interested in nitric oxide, and they said, you should make platforms that recognize this molecule nitric oxide. We're very interested in it from a toxicology standpoint for cancer, for on oncology, and they said it's an easy molecule to work with, so don't worry. And that was not true. It's not true. Nitric oxide is a very difficult molecule to work with. Of course, you may be familiar with the Nobel Prize story. It was an unidentified agent for a long time. Nitric oxide its role in the body continues to be very complex. It's very daunting for an engineer like myself. If you search for nitric oxide and cancer, you will get 10,000 papers, and 10,000 papers tell you that in your lifetime, in your career, you won't understand this literature. So it's why is it so complex? So nitric oxide is a small molecule signaling agent in the body. It's understood. The hypothesis is that its concentration in the body influence its physiological role. So when the concentration is low, it has a chemoprotective effect. It's a signaling molecule. It's used by the body as a it can diffuse several cell distances. It's a vasodilator. It's used as a neurotransmitter in the brain. And so it has these natural biological functions. And then the hypothesis is that in pathophysiology, if the concentration increases, then it can form these daughter products. It can form peroxynitrile. It can lead to oxidative stress, nitrosative stress. So this molecule, like most radicals, this is a familiar story if you work in free radicals in biology. It really has a Dr. Jekyll, Mr. Hyde type of thing. It has a nasty part, and it has an essential part. And it's the concentration. The hypothesis is that the concentration is what switches between these two roles. We have very poor ways of measuring nitric oxide in the body. And so early on, about a year later, we came up actually with the first optical sensor for nitric oxide. It's the first reversible optical sensor for nitric oxide. There are dyes that existed, but they bound daughter products. And actually, you can see here, this was the first example of CoPhMoRe. You can probably tell what we were trying to do with this diamino functionalized dextran. So this is a dextran. It has these diamino groups. We were trying to irreversibly bind a daughter product, but actually this system will actually reversibly bind nitric oxide, and it will cause a bleaching. You see here, it will selectively quench with nitric oxide, but not these confounding daughter products, these other complicating products, not hydrogen peroxide and so forth. So this was a big advance. We published this in Nature Chemistry, completely reversible. It's not, this is in a dialysis bag, so it's kind of hard to get complete reversibility, but otherwise it's not a covalent interaction, this very reactive molecule. And you can immediately use it in cells. This is macrophages. You've got carbon nanotubes in here. They are not activated by the carbon nanotubes. You can activate them separately with like adding lipopolysaccharide. You see here, you can easily image. You can get spatial temporal imaging of nitric oxide, and you see it's a very clear. If you add nitric oxide or you activate the macrophages, you can detect with spatial and temporal precision the nitric oxide. Carbon nanotubes, like many emerging nanosensor platforms, have the ultimate detection limit. This is something I want to emphasize, that we, not just carbon nanotubes, but we now understand in nanotechnology, when you shrink a nanosensor down to a small enough scale, your detection limit falls as the cube of the characteristic length. So you eventually reach a point where there's no concentration below which you can you are unable to detect. So this is a single diffraction limited carbon nanotube. In here is a rod, but you're seeing diffraction. I'm blowing up the pixels, and you're seeing here. This is, if you look closely, you're seeing stochastic. Those stochastic steps are individual nitric oxide molecules adsorbing to the surface. You're actually seeing at room temperature in a big messy environment, no quadrupole mass spec trap. I mean, just on a microscope slide, you're seeing single molecules of nitric oxide detect and cause a stochastic change. So this has been well documented. Now we have mathematical models that can turn these stochastic steps into concentrations and so forth. It's a very, very powerful approach. These nanosensors are giving us enormously selective interfaces to detect these signaling molecules. And this is part of a study we published in 2014 where we looked at two different kinds of sensor interfaces. You can tell me if I'm running out of time here. We implanted a nitric oxide sensing hydrogel into peritoneal, but also into tissue in the hindquarters of an A129 mouse. We also did tail vein injection of our sensors and looked at their biodistribution. And one of the things that we found, this is a DNA oligonucleotide wrapped around the carbon nanotube. If you just use this DNA oligonucleotide, it's very selective for nitric oxide. It's brighter than our previous Nature Chemistry paper. So that's why we switched to this oligonucleotide. It's your starting fluorescence intensity is higher, and the selectivity is pretty good. And however, if you do tail vein injection, you see that you immediately get carbon nanotube deposition along the left and right tail veins. So you see this here, and then nothing circulates. If you pegylate, it turns out if you pegylate the corona phase, you change the selectivity somewhat, but then you get complete clearance through at least the vasculature of the tail. So this is a familiar story to many of you in the audience. We went and did gel electrophoresis to show what was happening in the presence of serum. You're getting an absorbed protein corona phase with just the DNA. The PEG does a pretty good job of keeping this aggregation of proteins off, and then you're able to get clearance. And we've been using tissue spectroscopy where we use a liquid crystal tunable filter. Now this is becoming increasingly popular now. It's called hyperspectral technology, but it means that you get a 2D image, but at every wavelength. So we call these 2D lambda stacks. So it's a 2D image at each wavelength. You scan through the wavelength using this tunable imaging filter. And although it takes a little bit longer than a traditional imaging dark box, it allows us to subtract the carbon nanotube feature precisely. So there's no ambiguity. I'm going to show you a lot of images that look like we've doctored it, but actually it's just an overlay between the mixed image subtract out. You do a deconvolution of the carbon nanotube signal. Again, you have this in wavelength space. There's nothing else in the background that looks like a carbon nanotube, and then you can map that directly onto the optical image, the transmitted light image. Okay, so you get images like this, and we're absolutely certain that they're a carbon nanotube. And so you get maps like this. It's a very powerful technique. You can look at, these are actually from the A129 mice. This is looking at accumulation in the liver. We actually see that we get over 30 minutes, so the first thing that's surprising is that the pegylated version circulated just fine without any acute toxicity. It clears the lung, which is where many nanoparticles get held up, clears the lung, goes through the heart twice, and you do get, ultimately, it's not surprising to this audience, you get accumulation in the liver. That accumulation has a maximum, and then there seems to be an excretion. However, we were not able to detect above our detection limit carbon nanotubes in the urine or in the feces. There's some autofluorescence in the feces. It was short-sighted of us. The kibble that the mice were using had an infrared component that caught us off guard. I don't think that's discussed much in the literature, but still, so we saw this maximum of 30 minutes. What we decided to do was show for the first time that you could use these optical probes actually inside the living mouse in the liver and detect nitric oxide spatial and temporally in the liver of the living mouse. This is a huge advance for nanosensors. And so at the time, so this is what we did was we used this, what's called an SJL mouse. It's a very well-studied nitric oxide. If you've done any work in nitric oxide, you probably have used this SJL mouse. It's really a workhorse. You transplant lymphoma and you'll see, and that induces in the mouse a sharp respiratory burst from activated macrophages in the spleen, liver, and lymph nodes. It's a very reliable nitric oxide response. So it's used often to look at nitric oxide physiology, and there's a huge literature on this. This came from Jerry Wogan's work here. It was his. Okay, so you can look at the liver in situ. You can look at the excised liver. In the control mouse, you get plenty of fluorescence from our near-infrared sensitive nitric oxide sensor. If you as soon as you add the lymphoma, you get this nitric oxide burst. You see this quenching. It's very clear. You see this quenching in the liver. But then as a positive control, if you then excise the liver, the nitric oxide has gone away, and you see the fluorescence will come back. It's a reversible sensor, and it operates in vivo. And we showed other things too. So for example, there's nothing that limits the lifetime of a sensor like this. So this is actually an area of the lab that we've been working on for a while. We're making these tattoo diagnostics. These are hydrogel tattoos that we implant into thick tissue. And we're working on glucose, and also we have a project on insulin. But you can put these into tissue. These are hydrogels, CoPhMoRe wrapped carbon nanotubes, and you can do detection in real time of things like glucose and insulin. We're also working on cortisol. We have a project looking at cortisol. Anyway, this is data from our paper. This is we did histological sectioning at days 4, 180, and 400, and did not see any inflammation around this hydrogel implant site. These again are our A129 mouse. This is also in the same 2014 paper. And you see that the key here is that we decided to take this study out to 400 days on this population of mice. The sensor works just fine. You can actually see we did a small incision and implanted the sensor. Immediately after incision, there's enough nitric oxide produced from the macrophages that you see a quenching. And then 4 days later, you see the fluorescence comes back. It's a reversible sensor. And then we monitored this over more than a year. And it's just a huge amount. I believe it's the longest and implantable sensor has ever been put in a living thing. And it's like, and I've said that to a lot of audiences now. So if you kind of dig and find, if somebody can find another sensor, put 400 days, inside of a living animal, chemical sensor, then please bring that to my attention. But it really highlights the possibilities here. We have CoPhMoRe to detect possibly anything that we want in the human body, and we have a platform, we have a lifetime that could maybe take this out to, really we're thinking about a permanent sensor, a sensor that can monitor over weeks, months, and years is actually credible now. So that we're very excited about that. How am I doing on time? I think I'm losing a little bit of the audience. I'm sorry for the delays. Do I have until 1:45 or 10 minutes? All right. I'll try to tell you about our work in plants. Very excited about this. This is a huge growth area of the lab. So we understand a lot about nanoparticles in the human body because pioneers like Bob Langer but also people at Harvard, they recognized long ago that nanoparticles have an ability to transport drugs in the human body. So our knowledge of how nanoparticles interact in the human body is far superior today than plants. Plants have been completely neglected. There's a handful of papers, people thinking about maybe environmental toxicology. Plants are physiologically very different. And we became interested in plants by looking at the chloroplast. Okay, so the chloroplast, is a plastid. It's where photosynthesis occurs. It is, as an engineer, it was interesting to us that you could easily extract chloroplast, actually. I did not know this. You could take the spinach from your salad. You do very simple Percol separation. You do a centrifugation. You grind this up. You can extract these chloroplasts. As an aside, there's, from an engineering perspective, there are very few engineering studies that use the chloroplast. Just use it as a material, it performs a very interesting chemistry today. It turns CO2 to glucose using sunlight. There are chemists today all around the world tearing their hair out to do that kind of chemistry. And here we have this, you can just extract from the salad in your lunch these chloroplasts, and they will do this chemistry. And the key is that if you look in the engineering literature, essentially there's nothing. There's a study from the 1980s where people did the first electrochemistry on chloroplast, but then otherwise it's completely open. So one of the things that we found, I'm kind of skipping ahead a little bit, one of the things we found is that depending upon what you wrap a nanoparticle in, you'll do something that supposedly is impossible. And I've had plant biologists email me after my paper has come out and to tell me that what I'm seeing is impossible, and they tell me that I'm wrong actually. But actually, so what happens is you have nanoparticles. These are DNA wrapped carbon nanotubes They're negatively charged. They're flowing past an extracted chloroplast on a microscope slide. And what you're seeing is an irreversible accumulation through the lipid, the double lipid envelope that encapsulates the plastid. So the chloroplast from a plant biology perspective is like Fort Knox. Okay, nothing really gets through it. It's almost impossible to put a gene cassette in there, and it's and there's no endocytosis. There aren't these other active transport mechanisms that you have available from mammalian cells. So this observation requires an explanation. How it is so easy. You just take these parts. Not all particles will do this, but if you flow them across the extracted chloroplast, you'll get an irreversible accumulation. It gets even better. My postdoc Juan Pablo and we showed that you can actually deliver nanoparticles into the living plant, and they'll actually traffic and make their way into the plant cells. So here we're introducing them into the mesophyll, into these air spaces where there's fluid in the leaf, and then the particles will traffic and find their ways into plant cells. They'll actually go into the chloroplast even in vivo. So you're supposed to see here, this is chloroplast fluorescence. I'm really sorry, I can't get used to this. This is chloroplast fluorescence, and you're seeing co-localization with with carbon nanotube infrared fluorescence. Okay, so even in vivo, you'll find irreversible trapping of nanoparticles of a certain kind inside the chloroplast. So this opens up a lot of possibilities. So we first studied carbon nanotubes, and one of the things we found was that it didn't matter what the charge was, but it had the magnitude of the zeta potential had to be above a certain threshold in order to see this effect. So these are DNA-wrapped carbon nanotubes are negatively charged, and you see them go into the chloroplast. However, PEG-SWNT will not. So you don't see them here. Here's the chloroplast. No carbon nanotubes here. Zeta potential is only weakly negative, right? Chitosan, yes, even though it's positively charged, they still accumulate. And then this was particularly telling. This actually was very informative. Lipid wrap SWNT. If you wrap SWNT in the lipids that make up the lipid envelope of the chloroplast, you do not see transport into the chloroplast. This was a very important clue. With a lot of sleuthing and thinking, we came up with a mechanism that's quantitative. It's a predictive mechanism. We think it'll be very useful for the field. This is what we understand to be happening, okay? And I'll just show you the cartoons, and then we put this to mathematical modeling. Okay, so what we think is, it doesn't matter what the nanoparticle is. If its zeta potential is either strongly positive or strongly negative, it necessarily has an ionic cloud around it. Okay, we all agree on that. The bigger the charge, the more the ionic strength will form a concentrated cloud around it, right? That makes sense. As this ionic cloud approaches a lipid bilayer membrane, there's a very known effect that happens. It's been very well studied. If you increase the ionic strength around a lipid bilayer, you soften it. And people have done mechanical studies. They do AFM. It's a very well-known effect. So the first thing that happens as this particle approaches is the ionic strength will soften this lipid bilayer. Its effect is very well known. It's very quantitative. People know it quantitatively. I have mathematical equations for this, mathematical equations for this. This is what we presuppose. We suppose that what happens is that there's a lipid exchange, that there are unsaturated sites along the nanoparticle that exchange with the lipid. The lipid exchanges in complexes with the corona phase. As it does so, it will bring the nanoparticle through the softened lipid layer. What emerges through the other side is a lipid complexed nanoparticle, okay? It no longer has the driving force to go back through the membrane. Because one of the things that this mechanism has to describe is the irreversibility, okay? So now you have trapped inside the chloroplast, you have a lipid complexed corona phase nanoparticle, okay? And that explains why, at least for a period of time much longer than the entry, you have particles accumulating inside the chloroplast. If you put all that to equations, like in a longer talk, I actually go through the derivation, okay? But for this audience, you can take my word for it. You put all that, you get an equation, and the equation tells you the threshold, the zeta potential threshold as a function of size. That's the blue. Those equations show you that the entry is degenerate in zeta potential. There's a positive and a negative. That's our observation. So now take a look at this. So we derived this equation, and then we said, well, our theory says that any nanoparticle, as long as the zeta potential is above a threshold, will show this effect, okay? So that means it doesn't matter that it's carbon. It's just, and this plot is kind of a master plot that shows this, okay? So yes, carbon nanotubes are on the other side of this blue. So here's chitosan. So these we already had. But nanoceria, okay, it predicts that it will not do this. However, if you have, these are gold nanoparticles that you can buy commercially. Their zeta potential, they're larger, but their zeta potential is high. So and they do go through. But quantum dots, the zeta potential is not as large. Streptavidin encoded quantum dots, and they do not do this irreversible trapping. So this, I think, is a good direction for our field. We have an equation that makes predictions, and the predictions, it just so happens that they're able to falsify. They survive falsification, and so this equation involves dielectric constants of the lipid of the membrane in the water, the size, the inverse Debye scaling. There are membrane parameters that are well known. So these are known from lipid bilayer membranes, the membrane tension and the surface energy. We can look those up. This is an empirical parameter that we're learning about. It's the enthalpy change on lipid exchange. Okay, so it's the one aspect that we can't predict, but that one parameter can fit a whole family. So this lipid exchange. So we think this is a big victory. Now, just in the remaining minutes, for our paper in 2014, we did some flashy things. One of the things that you can do is you can put green light absorbing nanoparticles into the chloroplast. These are actually nanotube nanoceria complexes, and we actually showed two ways you can boost photosynthesis. So you can actually put them into the chloroplast, they absorb more light, you boost photosynthesis, and we call this a prosthetic photo absorber. It does not have any applications from what I understand. It's not the photosynthesis is not the bottleneck as to why a plant grows or this, but it is. But we're trying to illustrate that now you can hack into the plant in a way that you could not before. This is a paper that we published just at the start of this year. This is actually an explosive sensing plant that actually talks to your cell phone, actually. So here we're trying to illustrate some of the things. This is an area we call plant nanobionics. And the idea is, can we use nanoparticles to modify living plants, giving them functions that they don't have already? And as far as I know, we're the first to think about the plant itself as a platform for engineering. So like from an engineering perspective, the plant is very attractive. It has its own fluidic network. It powers it itself. It has access to its own chemical fuel. It generates its own energy from photosynthesis. It self-repairs. And so can we think about what a plant can do? And so this is supposed to push those limits. And we have a variety of more examples. We have a lot more examples coming down the pipeline that are even more sophisticated than this. But this is what we did here is we put a pair of sensors in. This is from previous work. This is actually a nitroaromatic sensing CoPhMoRe phase. This is a 7.5 tube. You're supposed to see here, it's not completely selective, but in a panel of nitroaromatics, I don't know why I keep pointing to the screen here. So here you see, you actually, you'll see a response to picric acid, but also a lot of other nitroaromatics, di and trinitroaromatics are recognized pretty selectively from this Corona phase. And this is a 7.5 tube. This is a 13.3 tube. You see here, there's a different panel of nitroaromatics that are detected on the 13.3 tube. So we actually published this in PNAS much earlier in 2011. However, what we did here is we inserted an infrared control reference and a sensor signal side by side inside the plant mesophil. So what happens is, if there are explosives in the groundwater, the plant will bring these up through the roots, through the stem, into the mesophil, where we have put a pair of sensor and reference nanoparticle spots that are in communication with a user's cell phone. So you can see here, you're adding picric acid. You wait 10 to 12 minutes or so. You can do this with a Raspberry Pi. This is a very low cost. It's the same kind of optics and things in your cell phone. And you're able to see, you're supposed to see here, there's a quenching signal here, a reference signal here. These are actually PVA wrap tubes. And then the plant will send your, we'll send you an email saying that it detected explosives in the groundwater. So you can go and take a look at our paper. We work out a lot of the engineering of this, and it's really our first stage of trying to modify plants and think about giving them new functions that they don't already have. And with that, I'm going to thank my group. I really apologize for going over. You know, I'm happy to answer any questions that you might have. Yes. Okay. Mm-hmm. I'm going to repeat the question, if you don't mind, so that the people who are online can hear. I don't know if they had your audio. So the first question is, have we tested these corona phases and the molecular recognition in more relevant media that are difficult, like a plant, like serum and so forth? So our early work, we were fascinated by just the structure of this molecular recognition. So our 2013 paper, we're just looking at recognition in buffer, or just looking at recognition. We become interested in applications. Our protein corona work, okay, we actually showed that the fibrinogen detection works in human plasma, not human serum. I think you'll look and see we show that the detection works in human serum. And I mentioned that too, that albumin, even though it's a sticky molecule, it does not confound the fibrinogen. We're working on an insulin sensor that can operate and give you real-time insulin sensing data. This is a project sponsored by JDRF. That paper's in the works, but we absolutely have characterized the recognition in human whole blood. Surprisingly, it works. It works in that environment, in fact. And the in vivo data speak for itself. Of course, the sensor is working in the exact environment where you want to use it. It's in a way, it's surprising, and I want to emphasize that these corona phases are not all of them, at least. They're not fragile, and they're not they don't appear to be confounded by the presence of these others. In a way, in the sensor field, we almost always test as a next step. If you're going to do biological applications, we almost always test in human serum. And people have questioned that because the application is not always for use in human serum. So, for example, if you're going to implant into tissue, a more relevant would be like interstitial fluid, which is not serum. But nevertheless, I think the idea in the field is if your sensor survives the aggressive conditions of human serum, then it'll survive these other environments. Ultimately, though, the proof is a sensor has to be used in the environment for its intended use. So our plant sensors, we test them out in plants, and if it goes into a mouse, we test in the mouse. It's the only data that really matter. So your second question is a good one for the people on the web. It's do the presence of the nanoparticles, I think we're going back to the dopamine imaging, the presence of the nanoparticles at the surface of a cell can change the function of the cell. So to that, I would answer in the following way. So first, that the nanoparticles themselves do not actually have to come in contact with the cell itself. That we are free to coat the surface, like we could put polylysine or something onto the surface. As long as dopamine can diffuse through that layer, it can excite and interact with the underlying cells. The PC12 cells are used often in the neuroscience literature. They're used to test out methodologies. They're already, when they're sitting on a surface, they're already fairly far removed from the in vivo environment. However, we are conducting studies now that involve intact brain slices and also going in vivo with some of the technologies that we're developing there. So your point is well taken. We have to think about what the nanoparticles do as a perturbative probe, right, and putting them in. However, I argue that they can be less perturbative than some of the tools that we have now. What do we have now? We have electrodes. You know, we can put those into the brain. We have genetically encoded fluorophores. I mean, these things are very invasive, right? So your point is well taken. But optical means also have an advantage in that the information itself can at least pass through tissue, and so you don't actually have to contend with getting a signal through an electrical conduit. So anyway, it's these are good questions. Did you want to take one from the web? Okay. Okay, thank you very much. Thanks for staying longer. Thank you.
Sorry for the juggling.
That's okay. It happens. Seems to happen all the time.
For talks like this, we have